Research Society for the Study of Diabetes in India, RSSDA, Tamil Nadu chapter, has immense pleasure in honoring Professor Sandeep Kumar Mathur. He is a distinguished senior professor and head of the Department of Endocrinology at SMS Medical College and Hospital in Jaipur, India. With an academic background and extensive clinical experience, he has established himself as a leading expert in molecular genetics and clinical endocrinology. His research primarily focuses on hormones and metabolic disorders, with a particular emphasis on the unique characteristics of the Asian Indian population, notably the thin fat phenotype, which has significant implications for diabetes and cardiovascular health. Throughout his career, he has been recognized for his contributions to the field of endocrinology, receiving numerous accolades, including the prestigious Commonwealth Academic Staff Fellowship and the Merit Award from the Government of Rajasthan. His commitment to advancing medical knowledge is evident in his prolific output, having published over 50 research papers in reputable journals. Additionally, he has been instrumental in leading various funded research projects that explore critical areas such as diabetes management, adipose tissue dysfunction, and inherited endocrine diseases, thereby influencing both clinical practice and public health policies. He is not only a dedicated researcher, but also a passionate educator, mentoring the next generation of medical professionals. His leadership in the department has fostered an environment of academic excellence and innovation, encouraging collaboration and interdisciplinary approaches to the complex health issues. A prominent figure in the medical community, he continues to advocate for improved healthcare strategies and patient outcomes, making a lasting impact on the field of endocrinology in India and beyond. Sir, the Tamil Nadu State Chapter RSSDA takes pride in honoring this great personality, Dr. Sandeep Kumar Mathur, with Professor M. Viswanathan oration on the 28th of September 2024 during the 15th annual conference at Chennai. I present this to Dr. Vijay Viswanathan, who is none other than the son of Professor Dr. M. Viswanathan, to do this honor.
We request Professor C.R. Anand Moses sir to present the orator with a medal. And we request Dr. V. Rajendran to enter the orator with a shawl and present a shield to him.
Probably this is the second time I'm chairing his oration. I remember in Ahmedabad 10 years back he gave the oration. At that time I was the chairperson. Now after 10 years I'm again the chairperson.
Special thanks to Dr. Mohan, who is not here, but introduced me with this wonderful team. Dr. Shangaman Velu, Dr. Shangungan, all wonderful people. And thanks to chairperson. You know, when we talk of diabetes, we always take a disorder of glucose metabolism. We talk of a disease where we talk there is a problem with the blood vessels. But here I am taking to a new horizon. A new horizon, the diabetes metabolic syndrome. Diabetes metabolic syndrome is primarily a disease of adipose tissue, and secondly, it is a disease, it's a genetic disease. You know, when the hormones were discovered, diabetes said to be endocrine disorder. Now the genes are being discovered gradually and molecular pathways are discovered. Diabetes and metabolic syndrome is in an era of molecular medicine and molecular genetics. So my journey started with my training from Imperial College in molecular genetics, and at that time I pursued so many ideas of Indian phenotype and started my investigations, and thanks to government of Rajasthan, they funded the first molecular genetic research lab in any endocrine department in the country, one of few in the country at that time. So, adipose tissue dysfunction and T2D interface, South Asian perspective. What I am going to tell you is, you know, what is adipose tissue dysfunction? An overview of the account is type 2 diabetes in Asian India associated with adipose tissue dysfunction, clinical, biochemical and radiological evidences. What is the molecular pathology of adipose tissue? And evidences on the role of pathogenesis in diabetes and metabolic syndrome in our population. How does the genetic susceptibility expresses in adipose tissue? To qualify diabetes as a disease of fat metabolism, a disease of the adipose tissue, adiposopathy, to establish as a clinical entity, I have to prove it, the genetic susceptibility to diabetes expresses through adipose tissue. Now, does the beta cell dysfunction reflect adipose tissue dysfunction? That is one of the original thoughts came to our own work, because if we say primarily adipose tissue, all other are consequence, then of course we have to establish that beta cell dysfunction reference adipose tissue dysfunction. Lastly, we took the model of disease, rare lipodystrophy syndrome, which is undisputed genetic disease with well-established adipose tissue originated pathophysiology which resembles metabolic syndrome. So we have generated data and they shared molecular pathway between lipodystrophy and Indian phenotype. And lastly, I will introduce in last slide the concept of adiposopathy, a primary disease with T2DM and metabolic syndrome, particularly non-obese person, as a clinical manifestation of that disease. So adipose tissue dysfunction. Adipose tissue is not only the largest endocrine organ, but it's a dynamic in the sense the whole adipocytes are replaced in every 10 years. It has tremendous capacity to hold excess calorie which comes from all your lifestyle factor in the adipose tissue, and it expands actually when challenged with positive calorie balance, and this is through a process of adipogenesis. Actually, I am emphasizing the term adipogenesis. If you ask me, my hypothesis, if you say current epidemic of diabetes, let it be in diabetes study data, everything I say is possibly, if we look into cellular molecular basis, is around the process of adipogenesis in this population. So adipocytes are recruited, mesenchymal stem are recruited when there is a positive calorie balance. They increase in number with the adipogenesis, and it is not associated, it is associated with angiogenesis, but not much of fibrosis and inflammation in a healthy individual. But if there is a failure of adipogenesis, it will lead to adipocyte hypertrophy, adipocyte ischemia because of not coupled with angiogenesis. It is associated with macrophage infiltration, which are inflammatory in polarity, that is on M1 macrophage and less M2 macrophage, then adipose tissue fibrosis, and this leads to a pathologic adipose tissue. So cardinal feature of pathologic adipose tissue are adipocyte hypertrophy, inflammation, oxidative stress, and hypoxia, and fibrosis. So this pathologic adipose tissue is associated with crosstalk with other organs. You know, hyper free fatty acidemia, ectopic fat deposition, inflammatory markers, adipocytokine, leptin, adrenaline, all they actually crosstalk with other organs like your blood vessel, atherosclerosis. There were controversy related to blood vessel and glucose and all. Glucose is, I think, minor player. More important player are inflammatory mediators as well as adipocytokines and free fatty acid. Then going to beta cell, it will lead to beta cell failure in pancreas and lead to impaired glucose, impaired insulin secretion and diabetes. And of course in liver, it will lead to insulin resistance, as well in muscle, it will lead to insulin resistance. This is the pathophysiology of adiposopathy, and it is reflected in, three patterns of body fat compartmentalization. At one end, there are individuals like lipodystrophy syndrome where there is a severe adipose tissue deficiency, adipose tissue dysfunction. So fat get deposited in your vital organs, lead to their dysfunction. At the other end are healthy obese individuals. They have tremendous capacity to expand the number of adipocytes with hyperplasia. So there is no deposition of fat in your vital organs like your liver, muscle, blood vessels, pancreas, and all those organs. And person doesn't develop any pathology. In between, there are individuals which we see our garden variety of diabetes and metabolic syndrome patient where there is an inability to expand adipocytes in number, lead to ectopic fat deposition, inflammation, adipocytokinemia, and your clinical disease. So this is the spectrum of compartmentalization. I already talked about this. Are T2DM metabolic syndromes associated with adipose dysfunction in South Asians? If so, what are the evidence at systemic level? So my journey in adipose tissue biology started when I started my first investigation after my fellowship in Imperial College in Genetics and Genomics, because Human Genome Project was not completed by that time. We were not aware what will happen in genomics because jivas were not a concept at that time. I started some intermediate phenotypic traits. What I observed in very simple study, that in T2DM patient as compared to their spouse, they had not only hyperglycemia, they had higher waist tube ratio. not higher BMI, they had hyperfree acidemia and hyperinsulin, high insulin resistance. But what importantly I could see there, that in diabetics there was a significant positive association with fatty acid level and insulin resistance, but not in the control. So I was sharing this word with my mentor T.J. Eitman. Tim, I observed it, only in diabetics FFA level show association of HOMA-IR, insulin resistance, but not in control. He said, Sandeep, it's a small window through which you can for a big science. And then I looked into genome and genome function, and the story became very clear why it's happening. Similarly, we find that systemic level diabetes associated with oxidative stress. And this another study, we participate in the Jiva of the India and that Indigo group, and we observed that inflammation, systemic inflammation marker, HSCRP levels, this is in 9,000 patient data, high HSCRP levels are associated with not only high prevalence of metabolic syndrome, but large number of components of metabolic. This is a very interesting aspect. HSCRP level, inflammation level goes up, not only the prevalence of metabolic syndrome increase, the number of components also increase. So it is, these are the observations, they open the window to look for another science. Similarly, what was observed that elevated HSCRP predicts risk of metabolic syndrome independent of obesity and insulin resistance. So if you talk what is insulin resistance, we never apply our mind beyond insulin resistance. But what I will say here, insulin resistance is an epiphenomena in my genome to phenopregnative adipose tissue directed dysmetabolism. Insulin resistance is an epiphenomena. It is not the root cause of all the problem. So this is what was shown in this work. So the inference is that T2D and metabolic syndrome South Asians is associated with heightened free fatty acid flux, systemic inflammation, and oxidative stress. What is the South Asian T2D and MET syndrome thin fat phenotype? Is it quantity, distribution, quality of the fat that matters? This landmark paper where, this is a review written by Ranjit from here only in Chennai, that Asian Indians when compared to Caucasian other races, and the data clearly showed they had relatively lower BMI, and they had predominantly central obesity or visceral obesity. We always say Indians are at high risk because of visceral obesity, because they have large volume or magnitude or amount of intraperitoneal fat. In fact, one landmark paper again from Chennai here from Dr. Mohan, he say that diabetics as compared to non-diabetics had larger volume of intraperitoneal or visceral fat. So we presume the Indians at high risk only because of large amount of fat present in the intraperitoneal compartment and large visceral fat volume. But you see, very simple experiments, surgically remove the momentum. There was no impact on dysmetabolism. So this theory was actually, one answer to so many questions. Remove momentum, no impact on your dysmetabolism. So what is role of different adipose tissue compartment high insulin resistance in Indian diabetics or type 2 patients? So we did a very simple experiment, look for body abdominal fat MRI fat distribution and any relation with insulin resistance. What we find that diabetics were associated with not only larger quantity of all these components, but not subcutaneous fat, but more important, but liver fat. Secondly, we find there was a strong correlation in diabetics, but not in the non-diabetics and non-metabolic syndrome patient. Diabetic and metabolic syndrome patient, it is the liver fat that showed strongest association with HOMA-IR. So this is something which expected. But what we look for, basically relationship between liver fat, subcutaneous fat, intraperitoneal fat, so-called visceral fat around the vital organs with the insulin resistance according to severity of insulin resistance. So this is something which was never done by anybody else in the research in this field. What we find with increasing quartiles, highest quartile, there was a very strong correlation with liver fat and insulin resistance, and there was a trend toward negative correlation with your subcutaneous fat. So this suggests, and there was no strong relationship between intraperitoneal fat. So we have raised a serious question of so-called visceral fat theory of Asian Indian type 2 phenotype, and we find it is the increasing liver fat that plays a very important role in dysmetabolism. Similarly, I look at some of the well-established published data. This are Anup Mishra, they say that Indians have high intraperitoneal fat, but they did not mention about low peripheral fat amount. These are two slides. They have less, subscapular thickness was increased, but they did not talk about this tricep skin fold thickness. That was less. So we always talk about all or more intraperitoneal fat, more abdominal fat, more central fat. We don't talk about less peripheral fat. We look for their supplementary material. They did not discuss, not included, but one of their data, their limbs, Indians have less fat in their limbs. That is equally important. That was not much emphasized and talked about. Similarly, we did a very simple experiment. Pioglitazone is the drug that enhances adipogenesis. So we looked for body composition in individuals before and after pioglitazone, 3 to 6 month duration. What we observed that pioglitazone induced normalization of HOMA-IR insulin resistance were correlated with increase in peripheral fat mass. Peripheral fat mass increased by enhancing adipogenesis and HOMA-IR became normal in this treatment-naive type 1, type 2 diabetic patient, newly diagnosed. So this all indicates it is a poor adipogenesis in peripheral fat leading to ectopic fat deposition. We are replicating this work for good publication and trying to make it multicenter also. And that is the major player of Indian phenotype, not the intraperitoneal fat around your vital organs. So this I already mentioned. So does T2D metabolism show association with pathologic changes in adipose tissue? What are the microscopic evidences? Light microscopic evidence. Are type 2 or metabolic syndrome associated with adipose tissue pathology? Let me tell you about this one wonderful study. Actually, this is a Canadian study. They compare South Asians with Caucasians, and they find, as expected, after adjustment for age, sex, and BMI, South Asian compared with white Caucasian had higher waist tube ratio. That means waist is high, but hip is also less. That is not talked about. Fasting high insulin level, low HDL cholesterol, and lower adiponectin. They also find South Asians had more body fat, lower lean muscle mass, increased waist to hip ratio, and also more liver fat in adipocyte area. They had more liver fat and large size of adipocytes. And very important part of their study is that when they adjusted for the adipocyte area or adipocyte size, the difference between South Asians and Caucasians disappeared. This is a very important finding. That means it's a poor adipogenesis in peripheral tissue and subcutaneous compartment that distinguishes Asian Indian for Caucasians. So the whole story, we have so many papers from Manisha Chandalia and so many groups. Asian have this, they have more visceral fat, less peripheral fat, not much talk about. But all difference is only because of limited adipogenesis capability in Asian Indian. That's why I say, even in diabetes studies say we have one, I think out of 10, 1% is diabetic metabolic syndrome. In fact, there is something I am able to dig out and I will share with Dr. Mohan about that. It is basically adipogenesis failure. That is the responsibility for all the story we are facing in our country. Then there is another study where they look for high fat feeding in Asian Indian. They find there was a basically 5 days of high fat diet leads to impaired insulin stimulated glucose disposal Asian Indian as compared to Caucasian. So Indians have a limited capacity to hold the challenge of the excess calories or fat intake, but they did not find any pathologic changes in muscle tissue. They said in their discussion probably it could be adipose tissue, but they did not study adipose tissue. And one recent study Dr. Mohan shared with me, this is published in August only, they actually studied Asian Indian dysmetabolism parameters before and after weight gain. And what they find, at the baseline, South Asian men had less whole body lean tissue, more whole body abdominal subcutaneous adipose tissue, ectopic fat in liver, but not visceral adipose tissue. They again actually in lines with our own finding, it is basically not visceral fat tissue around, that is the major player in Indians. They also find with weight gain, they actually treated patients 6 weeks with at least high calorie and 5 to 6 kg weight gain was happening, was induced, and post-prandial insulin was substantially greater in South Asian participant. Insulin sensitivity decreased by 38% South Asian as compared to 7% in Caucasians. and HOMA IR increased by 160% South Asian as compared to 4% in Western. That means when you are challenged with weight gain, the degree of dysmetabolism happening is multifold higher in Asian Indians as compared to Caucasians. The another part of the story is that at baseline mean adipocyte diameter volume were larger in South Asians, and they had few smaller adipocytes and more large adipocytes. So Asian Indian as compared to Caucasian had adipocyte hypertrophy, that means reflection of poor adipogenesis. The most important part is that when they were challenged with high fat diet and weight gain, Asian Indians, there was no increase in adipocytes distribution size because they are already at exhausted capability. But in Caucasians, adipocyte size increase. So conclusion is that Caucasians have capacity by increasing the adipocyte size because they have large number of small adipocytes, they can hold more fat. So they don't develop this metabolism. Indians have already exhausted their adipogenesis capability. So they are no more able to have adipogenesis, particularly in diabetics. So whenever they are challenged with the high fat diet or modern lifestyle, this leads to ectopic fat deposition and dysmetabolism. So whole story is actually roaming around your dysmetabolism. So this study, actually we are replicating this study. We also compare adipocyte size in relationship with insulin resistance in different compartments. So actually adipocyte hypertrophy is predominantly present in peripheral compartment, and it was only moderate in your central compartment. So we find, what we conclude, that thigh adipocyte size was significantly larger in diabetics in comparison with non-diabetics, whereas such difference was marginal in the abdominal compartment. In thigh, adipocyte size also showed a positive correlation with HOMA IR, waist circumference and triglyceride level. If you ask me, our high waist circumference, high triglyceride level, high insulin resistance is because of large adipocytes in the peripheral gluteofemoral compartment rather than your abdominal compartment. So it's all suggests it is a failure of adipogenesis in the peripheral and subcutaneous compartment that leads to thin fat phenotype. It is not your abdominal compartment. And secondly, we find what are the genes which are showing association with adipocyte size in our work, and they enrich the pathway of adipogenesis, lipolysis, and adipose inflammation related pathway. So basically it is molecular level failure of adipogenesis in peripheral compartment and to some extent in your subcutaneous compartment, and limited way to your very minimal and no evidence that in the visceral compartment. This leads to all ectopic fat deposition and pathophysiology. Similarly, M1, M2 macrophage indicates inflammation, and we find, actually this is not published work from ICMR project, we have seen that in Indians, peripheral fat as well as central fat, inflammatory M1 macrophages are increased in number and M2 macrophages decrease number. So in both the compartments, macrophages show, adipose show inflammatory cell phenotype. So on light microscopy, T2DM and METS syndrome in South Asian is associated with pathology changes in adipose tissue corrected by adipocyte hypertrophy and inflammatory macrophage infiltration. It is more so in peripheral compartment. So we are coming with theory that this Indian phenotype is because of limited adipogenesis, limited amount of peripheral fat with limited adipogenesis capability. That is the main culprit, and that should be focused in treatment. What is the molecular architecture of dysfunctional adipose tissue in Indian? Actually, long back we hypothesized that transcriptomic profiling of adipose tissue, that is an omics is a molecular technology of various adipose tissue reports with microarray can reveal, because that time this was the technology of choice, can reveal not only the molecular pathology of dysfunctional adipose tissue, but can also shed light on the role in pathophysiology of diabetes mellitus. So we use the technology of microarray. These are the omics technology. Basically, for any cell, any snapshot time, you would say whole genome sequencing, whole RNA profiling, whole protein profiling, you do whole metabolite profiling. The holistic approach is taken, that snapshot data is generated, and we see its correlation with clinical phenotype of interest. So this is called holistic or system biological approach. So we adopted system biological approach of transcription profiling. It is an omics technology which is primarily aimed at universal detection of whole set of RNAs transcribed by the genome. And this I already mentioned, it's an omics technology. There are two methods. One is hybridization-based microarray and other is your sequencing method, RNA-seq. So we actually fortunate to have first molecular genetic research lab in endocrine department in the country, and we had a microarray platform. And what we did, we were funded by ICMR to prove this theory at the molecular level. We look for this clinical phenotype of interest. All diabetes related phenotype we could study, and we had done microarray analysis of adipose tissue in the same time, in the same experiment. And this is our workflow. We had patient clinical phenotyping, then took fat biopsy during surgery, then transcription profile was generated, WGC analysis was done to see what are the modules of co-expressed gene. You know, we have 23 gene expression data, and we were looking for the trend, what genes out of them show association with your phenotype of interest. So we call it WGC analysis, and this analysis we look for what are the molecular pathway they enrich. So this was our pipeline. And what we find that peripheral adipose tissue, there were 7 modules of co-expressed gene. You know, complex did always say 23 down their permutation combination, but our story narrowed down to only 7 modules of co-expressed gene. And interestingly, those modules of co-expressed gene showed association with most of the phenotype parameters of interest. That means the whole pathophysiology of type 2 diabetes, right from adipocytes to adipocytokinemia to clinical disease, show correlation only with 7 modules of co-expressed genes. So they are the units of this metabolism, and they enrich the known pathways of adipose tissue we derived from animal model, cell line, as other human studies. So we could establish the pathophysiologic role of this adipose tissue pathology in this metabolism of this patient. So these findings suggest a role of peripheral subcutaneous adipose tissue, pathophysiology T2D and shows pathologic changes corrected by adipocyte hypertrophy and upregulation inflammation-related pathway. Again, same pathology, adipogenesis failure, inflammation was seen. In another study, we find basically contextual hub analysis. We again find the peripheral tissue adipogenesis failure. That's a correlation with type 2 diabetes mellitus. So inference is that in diabetics, peripheral adipose tissue show molecular pathology. They enrich the pathways of adipocyte hypertrophy, inflammation. Its modulo co-expressed gene show correlation with diabetes and other related phenotypic traits, thereby suggest role of adipose tissue in the physiology of diabetes. Same analysis was done in abdominal, both the compartments, abdominal subcutaneous and visceral. So several modules of co-expressed gene showed both bidirectional relationship with this metabolism, very important. Subcutaneous, and they enrich the pathway of adipogenesis and inflammation. Same thing was seen in subcutaneous compartment also, and same directionality of relationship was seen. So what important finding of this study is that nobody compared visceral subcutaneous same patient sometimes their molecular pathology. We find qualitatively it is same. So it is wrong to say visceral fat is pathological and subcutaneous fat is not pathological. In diabetes, they are qualitatively the same. Only thing, modules of co-express and their number proceed, they are bidirectional. Some is causing, some are protecting, and their number probably matters. So does the genetic substrate, now coming the question, if we say adipose tissue pathology or adiposopathy is a genetic disease, now question, does the genetic substrate for T2D matter syndrome express in adipose tissue dysfunction? If so, it supports the concept of T2D matter syndrome as a complex genetic disease of adipose tissue, so-called adiposopathy. Because genes are expressed in adipose tissue, then it's adiposopathy. What are the adipose tissues? Genome to phenome pathways of metabolome and T2DM. So 2 jivas were for diabetes in our country. One was from Dr. Mohan, what has been this European group, and this was Indian effort with Dr. Dwapan Bhajan. I was one of the partners in this group. And what we find, the genes which are showing associated with diabetes in community, majority of those genes does not enrich the non-pathway of insulin action or insulin secretion. So that means there is something else. So what we did, we generated transcription profiling of various insulin responsive tissue, particularly adipose tissue, and we find, and we also developed network of actually diabetes susceptibility gene, and there was marked overlap between them. So what we realized, the Jiva genes related directly to insulin secretion because they are already known to be expressed beta cell, but they through collaboration with other genes to insulin resistance also. So we came with the theory that insulin resistance, diabetes susceptibility genes are expressed in adipose tissue in their network and they cause a pathophysiology. So that means genetic susceptibility is acting through your adipose tissue. So supporting the concept of adiposopathy. Similar, this RNA-seq study, we again identify 2,750 differentially expressed gene in adipose tissue, Also, we identify 65 microRNAs, which are the actually potential target for discovery. My colleague in CDR at Lucknow is looking at all of them as a potential new target for drug discovery in our country. And 8 of them are already known to be linked with diabetes in databases. We also inferred a network of transcription factor protein kinase intermediate protein connecting differentially expressed gene, and interestingly we find majority of them were mapped to the Jiva loci. So in this way, in this very small studies published in scientific report, we could shed light on the genome to pheno pathway. Genetic susceptibility lead to altered expression of regulatory molecules like your kinases, transcription factors, microRNAs, and they alter transcription profiling of adipose tissue, lead to diabetes mellitus and metabolic syndrome. So we have created a sketch of molecular genome to phenome pathway of T2DM and metabolic syndrome in our population. So similarly, GWAS for BMI were done in our group, and we find such genes which are expressed in subcutaneous tissues in the protective compartment. Similarly, GWAS for actually metabolic syndrome identified the genes which are primarily expressed and affect the process of adipogenesis. Even GWAS for metabolic syndrome in Indian population again coned down on the process of adipogenesis. Now again an important part, because we say genetic susceptibility expressed in beta cell and insulin resistance is environmental. But we challenge this question because we generated network of genes which are showing association with diabetes mellitus, HOMA-IR and HOMA-beta. So there was significant overlap between diabetes and HOMA-beta gene network, but minimal over a HOMA-IR network. So it could be because of the beta cells are actually genetically determined defect. But HOMA-IR network enriched adipose pathology related pathway that was expected. But more important thing we added this study, genetic association of your beta cell failure. The genes HOMA-beta network also enrich adipose tissue patho-related pathway. The genes in a community is showing association with beta cell failure, but what pathway they enrich are adipose tissue dysfunction related. It is something surprising. The beta cell pathways are not activated, which are otherwise known in beta cell physiology, but adipose tissue pathways are activated there. And we've observed that these genes are expressed equally in all these tissue. So the pathophysiology of beta cell failure basically primarily in the adipose tissue, and secondary it is in the beta cell. So lipotoxic theory is there. Now coming to one more work which is little complex to understand for clinician. We use machine learning model based insulin resistant metagene. We took so many data sets of adipose tissue, muscle and all, predominant adipose tissue, and we use machine learning model. I think I will finish in 2 minutes. That what are the metagene which are common gene by machine learning, and then we apply to a data set of diabetics and non-diabetics of adipose tissue transcription, and that metagenome was able to predict diabetes in 73% of the patient. That means primarily pathology is in the adipose tissue, even when the person who developed diabetes or insulin resistance. So it is 73% predictably support this concept. Now coming to last part of my presentation, lipodystrophy syndrome, primarily monogenic disorder adipose tissue. Why we choose lipodystrophy? Because their pathophysiology well understood, genes are well identified, they are all located in the adipose tissue, and their whole pathology primarily from adipose tissue. If it is so, that means type 2 diabetes and they share the common molecular pathway, that means primary pathology of type 2 diabetes metabolism also primarily is adipose tissue in origin. So they are the rare disease. They share common actually clinical phenotype. They shared common pathophysiology, adipogenesis or calorie handling and leading to ectopic deficiency, same pathophysiology there in those conditions. And the genes regularly for lipodystrophy are actually in the process of adipogenesis. And in fact, genes showing association with diabetes also associated with lower amount of BMI, glutathione fat mass, and lower adipose mass in peripheral compartment. So we generated the hypothesis, and I think I can skip this slide. So gene interaction network of lipodystrophy gene and transcription profiling overlap. We replicated this work in another study, and we find the same thing. There were 18% overlap between the network of lipodystrophy gene and type 2 diabetes peripheral compartment transcription profile network. And very important, lipodystrophy gene played a role in altered expression as a correlation with disease-related phenotype in diabetics not in the control group. And last and most important finding was we could individualize a patient on the basis of expression 1, 2, 7, 8 of lipodystrophy gene. So now the end of my presentation, this work is still to be public. This is accepted in Frontiers in Endocrinology. And Dr. Mohan and his team and we work together on this project. And we look for expression of lipodystrophy gene and look for the association with diabetic-related phenotypic trait. We find all lipodystrophy genes were downregulated in diabetic patients in all the compartments. So, more on than this, expression of lipodystrophy gene out of 55 is to only on basis of 12, which are enriched in our modules of co-express gene, we can predict if someone is having metabolic syndrome or not. So you take fat biopsy by tissue, by your needle, because that can be easily taken up. You look for the expression of lipodystrophy gene. On the basis of that, you can predict one son is diabetic or non-diabetic, only on the basis of expression of 12 genes. The complex story of diabetes and metabolism narrowed down to only 12 genes, and some of their modules of co-express gene. And so inference is that T2D and MET syndrome are functional disorders of partial lipodystrophy gene because their pathophysiology very clear. Adiposity dysfunction leading to diabetes and metabolic syndrome. Therefore, these findings support the concept T2D and MET syndrome could be clinical manifestation of primary disease of adipose tissue, so-called adiposopathy. This is very strong evidence in favor of adiposopathy as the primary cause of diabetes. And in conclusion, I am presenting here a concept of disease, adiposopathy, primary disease. Because it starts with genetic susceptibility to your advanced end stage of your diabetes with complications. And as compared to your concept of metabolic syndrome, where we see cluster of disease, diabetes, hypertension, dyslipidemia, this. It is primary disease of adipose tissue from which all these complications grow. And we have shed a light on its genome to phenome pathway. That is very important. Until and unless we establish genome to phenome pathway, we can't say it's a complex genetic disease. So we have generated at least initial data to support this pathophysiology because genetic susceptibility, altered expression of regulatory proteins and kinases and microRNA, they lead to transcription profile change and pathology in adipose tissue, particularly poor adipose inflammation, and that lead to your clinical disease like beta cell function, hypertension, and your dyslipidemia and atherosclerosis. So at the end, I want to say the concept of adiposopathy, a complex genetic disorder with well-different genome to phenome pathway, could provide a diagnostic frame to imbibe the molecular diagnostic. That is very important, imbibing molecular diagnostic in your practice. And identification of novel targets for gene and cell-based therapy for future cure of diabetes. Because this is a window which my mentor Tim Eitemans asks, look for big signs. Because this is a window where genetic manipulation in future can cure your severe metabolic dysfunction diabetes. And thank you for your patience hearing. And word of gratitude to my teachers, Dr. Baldo who taught me medicine and Dr. Eitman who taught me genetics, complex trait. And what I learned from them, the easiest thing a doctor can do is to treat a patient. It's all written in books. But the lifetime challenge for a doctor is to discover what is written in the books. And thanks to my team, my surgeons who provide me tissue biopsies, scientists work in my lab, bioinformatician, and many thanks to my technician and staff who is supporting this work. Thank you very much.
Thank you, Dr. Mathur. I think we should give him a standing ovation. Thank you, sir. There will not be any questions since it's a guest lecture, but we should thank him for making such a difficult topic so simple for us clinicians to understand. And a new terminology I learned today is adiposopathy. Thank you, sir.
Thank you, sir.